This video will cover foundational concepts number 1A, proteins and amino acid structure and function. We're going to dive into protein conformation and conformational stability, which include denaturing and folding, hydrophobic interactions, and solvation layer or entropy. So first, I'm going to touch on conformational stability. So proteins have a unique conformation, which is essentially its structure, which can be in 3D form, and it is highly related to its function. The protein stability depends on the environment and interactions, such as hydrophobic, hydrophilic interactions, hydrogen bonding, and cysteine bridges. Proteins will spontaneously fold into lower energy conformation, and environmental factors affect what the most stable form of the protein is. So if the protein is in an aqueous environment, which is like water filled, then the hydrophilic residues would be on the outside, so there will be polar on polar interactions. In a higher pH, the residues will be deprotonated, which we've seen in amino acids. And finally, in optimal conditions, they fold into their native state, which is the lowest energy and functional form, which can then be denatured or unfolded when the environment changes. The stability of this conformation can be described by delta G, which means the difference in Gibbs free energy. So the delta sign means difference, and G stands for Gibbs free energy between its various states of conformation, so folded or unfolded. Hydrogen bonding and the hydrophobic effect play significant roles in the conformational stability of a protein, which is dependent on the temperature, pH, salt concentration, and presence of chaperones, which are other proteins that assist in folding and unfolding. So ideally, the protein wants to fold into a state to maximize entropy, or the randomness of the system, and minimize intrinsic energy, because lower energy means that the protein is more stable. Each protein has a unique conformation, as it firstly comes from the sequence of amino acids comprising its primary structure, as well as the process of folding that it undergoes. So the folding process may be influenced by the solvation environment or different changes, like we talked about before, temperature, pH, and salt concentration. Protein folding refers to the process through which a protein is organized or folded into its proper secondary or tertiary structures. Essentially, folding brings the primary chain into a 3D structure. As protein form is highly related to function, protein denaturation most often results in a loss of function. So this folding process is very important to ensure that proteins will function how they are meant to. And many of the proteins will fold spontaneously, whereas others require the assistance of chaperone proteins. So sometimes the chaperone proteins may misfold the proteins, or the inappropriate conditions may lead to misfolding, which leads to an inactive or sometimes a toxic protein. And this is especially a problem because the most proteins are folded during the same time as the polypeptide sequence is still being translated. So it can cause many errors that may not be fixed. So as mentioned in the previous slide, to be biologically active, a protein must have a specific conformation under the proper physiological conditions in order to function. And like misfolding, a change in the conditions can also cause denaturing, which is the loss of quaternary, tertiary, or secondary structures as a protein will unravel. When it's been denatured, it can often be reformed when conditions are changed, and this may happen spontaneously where it will refold to the native states. Denaturing is essentially the loss of the native 3D structure so that the protein no longer functions. And it's most famously known as when a protein was denatured with urea. Many proteins cannot refold to their native state once they've been unfolded due to the high number of possible conformations that the polypeptide sequence can assume. Folding a protein can be tricky, and the chaperones may assist in folding the denatured proteins back into their native state. It's important to note that the protein denaturation does not disrupt the primary protein structure. So in terms of denaturation, I've just created a quick chart to help you see the different protein structures that are affected with each process. So enzymes are the only way that the primary protein structure can be broken down. This is obviously really important for us to digest foods, and it happens because of the hydrolysis of the peptide bonds. So in a pH condition, which could be either really high pH or really low pH, the potential effect is that ionic and hydrogen bonds are affected and disrupted, and this can change the secondary, tertiary, or quaternary structure. With a really high salt concentration, this can disrupt the non-covalent interactions, and again, disrupt every protein structure except for the first one. And with different solvents, hydrogen bonds can be disrupted. So later we're going to talk about something called SDS-PAGE, and that is a type of solvent that is used to break down sulfur bonds. And finally, different temperatures can cause spontaneous rearrangements at different melting temperatures. So, for example, when something is a really high temperature, it has higher energy, so it will have a different conformation than if the protein is at a lower energy. So the solvation layer is essentially the structured organization of a solvent, such as water, and a solute, and in our case, that would be a polypeptide or a protein. So when a protein displays hydrophobic residues on its surface, the surrounding water will orient in a highly structured way in order to optimize the number of hydrogen bonds among water molecules. This highly ordered arrangement has a much lower entropy and is much less favorable than if polar side chains were present on the surface of the protein. So therefore, the conformation, which pushes the hydrophobic residues into the inner core of the protein, leads to fewer disruptions of the water's hydrogen bonding and allows for less structure and a higher entropy, which increases the protein's conformational stability. So for way back to previous slides, remember we want high entropy, which is higher randomness, and in the ideal protein. Hydrophobic interactions use both repulsion and attraction, so a push or a pull, in order to contribute to a protein's conformational stability. This push comes from the thermodynamically favorable shielding of the hydrophobic residues when they are placed in the inner core of the protein. So as I said before, it's more stable when all the hydrophobic residues are in the middle of the protein rather than if they were interacting with the solvation layer. The pull results from van der Waals forces, which occur between nonpolar side chains on the polypeptide, especially London dispersion forces, which are amplified when the hydrophobic residues are really close together on the inner core, which produce a larger effect in larger proteins. So we'll talk more about van der Waals forces and London dispersion forces, but they are essentially present between all atoms. The hydrophobic effect is a consequence of these nonpolar and polar interactions. So in an aqueous environment or water-filled environment, the hydrophobic residues will be attracted to one another, while they will also be repelled by the polar aqueous environment, so the water and the ions. For this reason, again, the hydrophobic amino acids are going to be found in the interior of the protein rather than the outside of the protein. So I'm just going to go into a bit more detail about the differences when the outer protein residues are polar versus when they are hydrophobic. So when these outer protein residues are polar, the water molecules in the solvation layer can easily form hydrogen bonds in different arrangements. So the water molecules will be in all these different arrangements surrounding the proteins. However, if the outer layer of the protein is comprised of mainly hydrophobic residues, the number of hydrogen bonds that can form is extremely limited. So this requires the water molecules to be oriented more rigidly, resulting in an unfavorable reduction in the entropy. So you'll be able to see that in the image on the right, how all of the water molecules are arranged in such a way that they're just kind of like little open triangles around the protein. So that is what would happen with a hydrophobic protein. So this can also be explained from a thermodynamic standpoint, which we'll talk about more in later videos. The hydrophobic effect has to do with entropy. So yet again, entropy is the measure of disorder in a system, and you want to increase entropy, resulting in more disorder with time. So if a protein has these hydrophobic residues on the outside, then the surrounding water molecules have fewer residues to bind to on the surface of the protein. So this will reduce the ability of the protein to interact within the body. So as this video is pretty loaded, I've just created a list of some important terms and concepts that you should try to learn in more detail for the MCAT. I've made a set of Quizlet flashcards that are going to be available, and they will be password protected. So just stay tuned till the end of the video and you will see the passwords so you can log on and have access to them.